Honors Biology, third quarter review, part 2, transcription and translation. Remember that transcription and translation happen at the same time. We're talking about taking the codes from DNA and bringing them out to the ribosome in the cytoplasm to make our proteins. Proteins are super important. They're used for almost everything in living things. So the codes for them in DNA are also very important as well. If we're making muscle protein or cartilage in our ear or enzymes, all those things are proteins that are coded for by DNA. Now remember, RNA is very similar to DNA. They're both nucleic acids. However, RNA is different in 3 ways. RNA has ribose as a sugar. DNA has deoxyribose as a sugar. RNA has uracil as a nitrogen base instead of thymine. If you ever see uracil in an answer, you know you're dealing with RNA. If you ever see thymine, you know you're dealing with DNA. Remember that A in DNA codes for T in DNA and U in RNA. So all thymines are replaced by uracil. If you have a T in DNA, it still codes for A in RNA. So A codes for U in RNA. T codes for A. The third difference is RNA is single-stranded, whereas DNA is double-stranded. Transcription. You don't have to know step 1. However, the enzyme that you do have to know for transcription is called RNA polymerase. Easy to remember because RNA polymerase makes RNA polymers, and you make RNA polymers during transcription. RNA polymerase will bind to the DNA template strand and then bring in the correct nucleotides that are complementary to the DNA template strand. So here we have adenine coding for uracil and RNA brought in by RNA polymerase, guanine for cytosine, thymine for adenine, and so on and so forth until we get to a termination sequence. Once the termination sequence is reached, RNA polymerase detaches and the messenger RNA is done with transcription. Now there's one more thing that has to happen to the messenger RNA before it leaves the nucleus. We have to cut out the non-coding regions. The non-coding regions are called introns. These are cut out. The exons are the coding regions of RNA, and are the parts that are going to be spliced together and be used to make our proteins. So once again, once we are done with transcription, we're going to cut out the introns, splice together the exons, and now the messenger RNA is complete. So we're done with transcription. We have a copy of the codes from DNA. The genes that make proteins or code for proteins are copied onto messenger RNA. Remember, the reason why we're making messenger RNA is because DNA stays in the nucleus and the ribosome makes the protein. So the codes are here. The ribosome is here. The messenger RNA gives the codes to the ribosome so we can make our protein. And that's the whole point of the messenger RNA. It's a messenger of the genes from DNA. DNA is safe and secure inside the nucleus. The next step, messenger RNA binds to a ribosome, and the next step is translation. Remember, AA, translation amino acids. We're going to chain together our amino acids at the ribosome using messenger RNA. The 3 things needed for translation are messenger RNA, ribosomes, and transfer RNA to bring in the amino acids. So now we're going to talk about translation. We're done with transcription. We made our messenger RNA from the DNA template, and we cut out the introns. All right, the genetic code. We're going to be using the genetic code from the book. That's a little bit easier to use. But remember, the important thing you need to know is the genetic code uses messenger RNA, not DNA, not transfer RNA, messenger RNA. And if you don't have messenger RNA, you can't use the genetic code. The genetic code codes for all the amino acids in the protein. The way you use this particular genetic code is you go with the first base, which is in this case A. AUG is a codon consisting of 3 nucleotides. First base is A. Second base is U. So you just kind of go together, A, U, and then G. That codes for methionine, which is a start codon. We have a couple stop codons. Basically, those stop codons just tell the ribosome that you're done with translation. All right, so let's do a little practice. If DNA has a template strand of TAC, what's complementary in messenger RNA? AUG, T codes for A for U, remember there's no T's in messenger RNA, and C for G. Now the next question is what amino acids can be brought in. Now I'll always give you the genetic code. AUG brings in methionine. Now take a look here. We have methionine being brought in. We used AUG on the genetic code. However, there's another part, transfer RNA. Transfer RNA has an anticodon of UAC that's complementary to the codon AUG. This lets the cell know which amino acid to bring in. The ribosome is going to bring in only methionine because only UAC on the anticodon transfer RNA binds with AUG on the messenger RNA codon. They're complementary to each other. So there's a couple parts to this transfer RNA. And remember, transfer RNA transfers in amino acids. The first part of the transfer RNA is an anticodon, complementary to messenger RNA. There's a body of this transfer RNA, and then the third part is the amino acid, and that's the part that's being brought in. So let's see if you can figure out the next one. CGU. See if you can figure out what CGU is going to code for. And that's going to bring in arginine. Now keep in mind, if you try to use the anticodon on transfer RNA, GCA, that's not going to code for arginine. So that's going to give you the wrong answer. All right, GUA, if you want to double check that, that's going to be bringing in valine. So you should be able to go from DNA to amino acid using the genetic code. You have to figure out messenger RNA first. You should be able to go from transfer RNA to messenger RNA to DNA. And you should be able to go directly from messenger RNA to the amino acid. Remember, the anticodon is on transfer RNA, and that's complementary to the messenger RNA codon. A codon is a set of 3 nucleotides. So let's take a look here. This is AUG, and that's one codon bringing in one amino acid. 3 nucleotides on RNA brings in one amino acid. So if I ask you how many nucleotides are required to code for 5 amino acids, the answer would be 5 times 3, which would be 15 nucleotides. If I ask you how many codons are required to bring in one amino acid, AUG is one codon, and you need one codon for one amino acid. So 5 amino acids requires 5 codons, or 15 nucleotides. I always put a line in between my codons to keep them separate and not to get them confused. So let's see if you can tell the story. After I explain this, explain it to yourself. The first step is transcription. That happens in the nucleus. RNA polymerase binds to the DNA, brings in the complementary nucleotides, complementary to one side of the DNA molecule, that are going to be transcribed to make messenger RNA. Remember, we're transcribing a gene, which are the codes for a protein on DNA. After transcription is complete, replacing all T's with U's, we have introns and exons. We're going to cut out those introns, and we're going to splice together the exons. The exons are the coding regions of RNA. The introns are non-coding regions that we're not sure about what they do. The next step, after the exons are spliced together for messenger RNA, the messenger RNA leaves the nucleus and binds to a ribosome. The ribosome will attach to the messenger RNA, and then using the codons on messenger RNA, things like AUG, we're going to bring in an anticodon on transfer RNA to bring in the right amino acid, represented here. Once a transfer RNA drops off its amino acid, it detaches. Another anticodon on the transfer RNA binds with a codon at the ribosome, brings in another amino acid, and this process just keeps on repeating until we've translated the entire messenger RNA and made our entire protein. Remember, a protein is just a sequence of amino acids. This is how things like insulin and muscle protein, actin-myosin, are made inside your body. All right, at this time, I'm going to back it up a little bit and see if you can explain it to yourself, and repeat this part of the podcast if you need to. All right, let's move on. Evolution. The second part of third quarter dealt with evolution, or the change in genetics of a population over time, as a result of a variety of factors, the most common of which is natural selection. Charles Darwin was the first person to discover evolution by natural selection. You should know that. Darwin's finches. Darwin discovered a lot of finches on the different islands of the Galapagos. They adapted to their environments and selected different traits for different beak sizes as a result of the food available. Adaptive radiation basically means radiating from a central point and then adapting to your environments. Remember, this is how speciation occurs. The first step is reproductive isolation, isolated gene pools, and then mutations in this population here are not passed here, and mutations here are not passed here. Now, selected for mutations will result in differences in populations over time. So after enough differences accumulate, these populations will no longer be able to reproduce, and they're considered different species. We're going to skip this for now. You don't have to worry about this until fourth quarter. Wallace was the guy who came up with the idea of evolution by natural selection, identical to Darwin, but he came up with it later, so he wasn't given credit for it. But if Darwin hadn't published his work, we'd be studying Wallace's theory of evolution. Now, evolution wasn't come up with by Darwin first. It was actually done by Lamarck. However, Lamarck got the mechanism wrong. Darwin was the first to come up with evolution by natural selection, or nature selecting for traits that promote survival and reproduction. Lamarck thought that things evolved or changed over time as a result of use and disuse, acquiring traits based on your environment. Now, this is not how it actually works, but at least he was thinking about how things change over time. What Lamarck would say is that if you're in an environment where you need like a longer neck to reach the higher leaves on a tree, or perhaps sharper teeth if you're a tiger, or whatever animal you are that needs an adaptation, you would get it just because you need it or you require it to survive. This is not how traits actually work. Traits are coded for by DNA, and you're either born with the DNA for a trait or you're not born with the DNA for a trait. Nothing that you do during your lifetime is actually passed on to the generation. For example, if you work out your left arm and it gets really big, your kids aren't going to have all big left arms. But Lamarck would have said, yeah, you would have had big arms if you worked it out in your adult life. Now, Lamarck also thought that you lose stuff as a result of disuse. So he would say that something just disappears because you don't use it anymore. Now, things can disappear, like the eyes on a blind cavefish. However, that's the result of mutations that don't have a negative survival disadvantage to the organism, not just because you don't use it anymore. So what did Darwin say? He said that all living things have the ability to overproduce what the environment can support, basically exceeding carrying capacity. And as a result of that, some will survive and some won't survive. There's also competition. Competition for food, getting enough resources to keep that cell respiration going. For mates, reproducing, sexual selection. For nesting spots, if you're an animal that has a nesting spot. And escape predators. So competition for limited resources drives natural selection. Those that can get those resources are better adapted to survive in their environment, pass on the genes that allowed them to get to the resources, and those that don't are less successful in passing on those genes. So you put together variation and competition, and you get yourself natural selection. Natural selection is nature selecting for the traits that best promote survival or reproduction in the environment. It's also called survival of the fittest. Now, what is selected is determined by the environment. If you're in a cold environment, you're going to be having selection for things like thick fur, blubber, things that are going to help you survive. If you're in an environment in the savanna where you're being chased by things like lions, possibly speed and faster metabolism to survive predators, or different camouflage, hiding, things like that. Those are the things that would promote survival, and those are the traits that get passed on more frequently because the survivors are having the most children with those traits. So keep in mind, fittest are the ones that survive to reproduce. The dodo bird, not very fit, not from the sense of being out of shape, but because when the environment changed, when humans brought dogs and pigs, there was no adaptation in the genetics of the dodo bird that allowed them the ability to survive when we changed their environment for them. And they died out. If you don't adapt, if you don't have a genetic trait that can be selected for, and you don't migrate to an area that you are adapted to, then that's really your only choice. And that's why there's no more dodo birds. So survival of the fittest. Who's fit is determined by the environment. In a dark environment, a dark moth is more fit. It's hiding from those birds more easily. In a neutral environment, neither is more fit, and both are selected for equally. In a light environment, the light moths are more fit because they're blending in better and able to survive bird attacks. So you do need to be familiar with some observed examples of evolution, and the first one is the peppered moth. What happened was, in the early 1800s, in pre-industrial England, there was a lot of lichen that's light-colored growing on the trees, light environment, and selection for the lighter-colored moths. The industrial England, we had a lot of pollution, lichen on the trees, and as a result, we had the phenotype of the dark-colored moths being selected for and becoming the common one in the population. This has been recorded, and we have the data for this. In the mid-1900s, we cleaned up our pollution, and the light-colored bark lichen returned, and the lighter-colored moths were able to survive better because they blended in. This is called industrial melanism. The melanism is what gives the dark or light colors to the moths. Melanin is a pigment that makes the darker moths dark. And industrial means that we're talking about the selective pressure, what caused the selection to occur. When the environment changed, what was selected for changed.